There are trillions of cells in our bodies, and each one contains a parcel of important information. From the colour of our hair to our risk of developing certain diseases, the information that determines all of our physical characteristics can be found in our DNA. And there's a DNA molecule inside the nucleus of every single one of our cells. But stretched out, a DNA molecule is about 2 meters long, while cells, as we know, are very small indeed. So how does a DNA molecule squeeze itself into a structure 10 times narrower than a human hair? Well, just as you couldn't fit 10 pages into an envelope if you scrunched them up and stuffed them in at random, the DNA molecule fits because of the way that it's organised and folded. We have discovered that faults in the folding of our DNA can have huge consequences for our health. We know, for instance, that it can cause rare blood diseases. But we also know that if we can find the fault, it might be possible to fix it, and to eliminate the disease that it's caused. In the laboratory, this concept has been proven to work. The next step is to find faults that cause other, more common diseases, like diabetes, and see if they too can be corrected at a molecular level. For patients living with a disease linked to the way that their DNA is folded, a cure of this kind is still some way away. But with this significant first step, endless possibilities unfold. Join us at the Royal Society's Summer Science Exhibition.
In all honesty, we still really don't understand how the brain works. But we have, over the last 100 years or so, been able to being able to decode what the brain does. So the simplest description is that it comprises of billions of neurons. Each of these neurons can be thought of as a tiny switch connected with lots of wires to other neurons and switches. And by passing electrical information along these, it acts as a large electrical circuit which governs everything we do from touch to movement to vision to how we think and how we feel. There are a number of ways that you can detect these electrical currents around the brain. The most accurate one is to put an electrode straight into the brain at the place you want to listen into. However, this involves drilling into the skull and putting electrodes into the brain, and that's very difficult. Another option is to put electrodes on the scalp, so you just place them on there and measure the electrical potential on the scalp as an electrical current passes somewhere in the brain. But there's also a third way, and this is the way we're interested in. What you find is when an electrical current passes down a wire, it generates a tiny magnetic field. Now these magnetic fields are quite useful because they're not distorted as they pass through the skull, which means that if we can detect them, we can be quite accurate in locating where they came from. And it's these magnetic fields we're interested in.
MEG works by taking the naturally occurring magnetic signals in the brain, and from detecting these signals we can then identify where activity in the brain is happening. We use an array of sensors and we look at what each sensor can see and find where in the brain that happens, much like with radar, triangulating position and finding where signal's coming from. The current MEG scanners are so big because in order to detect these signals we have to use something called superconducting quantum interference devices. They need to be cooled down to minus 269 degrees, so they have to be kept in a vat of liquid helium. The new sensors that we're developing are something called optically pumped magnetometers. These are room temperature sensors, so we don't have to keep them cool. Also don't have to keep them far away from the head. We can put them directly on someone's scalp. So in order to keep the sensors on someone's head, we have to put them inside a sensor cast. And the way that we do that is we use a 3D printed one that's tailored particularly for the person being scanned. This is based on their MRI scan, so it fits them perfectly. When we only had a few sensors, we used this one, and that fitted just over a particular region that we were interested in. But then as we started to get more and wanted to explore more regions, we moved up to this hedgehog-looking one, which allows us to put sensors wherever we need them to be. The problem with it is it's tailored just for one person, so only that person can wear it. And another issue is it's quite scary, especially if it's a child that's going to put these on. They're not going to want to put a scary mask over their face. So what we did do instead is we've moved towards using a bike helmet. And by using this, it's something that children are familiar with. They're more willing to put on. So using these new sensors where we can bring them into the scalp, fit them to whatever head size we want, we can start to look at how a child's brain develops. This will hopefully let us see how certain disorders come about, such as Asperger's or ADHD, several things that develop in childhood.
We can use brain scanning like MEG, for example, to tell us about mental health by building up a picture of how the healthy brain functions using a really large group of people. And then we can use this to compare with groups of people with specific mental illnesses, and we can see where these differences lie and exactly what differences are occurring in the people with mental illnesses. The advantage of the OPM system means that we can do experiments that we weren't able to do previously. And we've developed it up to the point where we are able to use the OPMs to measure people when they're moving their heads around, doing kind of almost sporting activities like bouncing a ping pong ball. That's something that we couldn't do with the other previous MEG system. There's the potential for using an immersive environment such as virtual reality in order to see how the brain is changing in a more naturalistic way. And we can then use this information to perhaps tailor our therapies accordingly to help people more in their day-to-day lives.
Inside this tube is a tumour sample. Tumours are more than just cancer cells. They also contain healthy cells and molecules that support the tumour. This is called the tumour microenvironment. And this sample is being broken down into all these different parts, so scientists at Bart's Cancer Institute in London can study them. This forms part of the ambitious CANBUILD project, with the ultimate goal of growing mini tumours in the lab to help understand the disease and develop new treatments.
In the past, we've tended to grow cancer cells on their own, just on plastic, but the body isn't plastic. And so people around the world, including the CANBUILD team, are trying to build human models of the tumour microenvironment, which contain just not the malignant cells, but other cells in a more relevant three-dimensional way. And what we decided to do was take apart the tumour that we wanted to study. Now, of course, if you take apart a watch or a plane or a car, it doesn't necessarily going to tell you how to put it back together again, but at least it gives you all the working parts.
Step by step, the CANBUILD team, made up of biologists, engineers and other scientists, carefully extract each different type of cell. They're studying 4 important properties of both the cells and the complete tumour tissue. They're analysing the architecture and mechanics of an intact tumour sample. They're studying the molecules that send signals between the different cells and the proteins that support the tumour. They're locating and counting immune cells in the tumour sample, which is helping them understand how the immune system recognises cancer. And using specialised computer software, they're studying the faulty genes in the patient's tumour and how they affect its growth. As they figure out how each part of the tumour works, the team is now starting to build artificial tumours in the lab.
My dream is that we succeed in making a human cancer model that has all the relevant cell components, but also just doesn't sit there for a day or so, but actually evolves and changes, and that we can usefully use that to find new treatments.
Malaria is one of the most ancient diseases of humanity, affecting people for as long as we know, and it still kills hundreds of thousands of people every year. Every few minutes a child dies of malaria. By the time this film has ended, there will have been another death. This could be prevented if we had a vaccine that protects people against malaria. Vaccines can wipe out diseases. Smallpox is gone and polio is almost defeated. It is now our quest to develop a truly effective vaccine to prevent malaria. Malaria is caused by a parasite that is injected into our bodies from an infected mosquito. The parasite lives inside the human liver and then the human blood, where it grows and divides. While in the blood, it causes the sometimes deadly symptoms of malaria. To defend us against invaders like malaria parasites, our bodies make antibodies. Antibodies help our immune systems to detect and clear invaders before they make us ill. But the malaria parasite has a solution. It gets inside human blood cells, where it hides from antibodies and multiplies in secret. An essential part of the machinery that the parasite uses to invade blood cells is a molecule called Rh5. This binds to a molecule in the red blood cell called basogen. Antibodies that stop Rh5 from binding to basogen leave the parasites trapped outside the red blood cells where they're destroyed. Vaccines contain pieces of invaders. These train our immune systems to make antibodies. If we know which part of Rh5 is targeted by the most effective antibodies, we can use this as a vaccine. This will prepare the body to make the best antibodies, stopping us from getting malaria. To do this, we needed to work out exactly what Rh5 looks like. For this, we used a huge machine called a synchrotron. The synchrotron is used to fire a beam of X-rays at a crystallised array of many copies of Rh5. The way that the X-rays are deflected when they hit the sample allows us to work out what Rh5 looks like. At the same time, vaccine trials took place in Oxford, injecting a vaccine containing Rh5 into volunteers, causing their bodies to make protective antibodies. We discovered that the antibodies that bind to two key parts of Rh5 are the most effective. We are now making a new vaccine containing these parts of Rh5. Humans immunised with this vaccine are expected to make the highest quality antibodies. Our improved vaccines will be tested in controlled clinical trials in volunteers. Those that work will be tested in areas of Africa where malaria is a major threat. Our dream is a vaccine that will help many millions of people throughout the globe. We hope that our studies of Rh5 are allowing us to take an important step towards this dream.
So let's say a patient comes in with pneumonia. Obviously we'll hear the patient's experience and see what's been going on, listen to the chest, do a chest x-ray, and that will tell us that maybe there's a severe infection in the lungs, but it won't tell us what the cause of it is. So we'll know from previous experience that it's maybe one of a number of 5 or 6 suspects, and we'll treat the patient with antibiotics that would cover most of those suspects. But what we'll also do is do a blood test, and we'll hope that the bacteria in the blood test we can grow in a lab, and that will tell us which antibiotics are going to work. Now what the research group is uses an entirely new technique where they break open the bacteria and extract the DNA.
Tuberculosis in particular is a slow-growing bacteria, so it can take up to 6 weeks to grow each one individually to see whether it's resistant to any of those drugs. And the benefit of performing whole genome sequencing is that you have your sequence very quickly within a matter of days, and from that you can work out which antibiotics it will be resistant to.
So in the old world you're doing a dozen different tests, each of which costs money. In the new world you sequence the genome of the bacterium, and from that you derive all the information you need to answer all of the questions that you want to ask. What species is it? Which other TB samples is it related to? Which drugs is it resistant to, and which will kill it? And it's just one test, and it costs about the same amount of money as one of the many other tests we currently use.
It's extraordinary that this is all immediately available, and even just 10 years ago it might have taken you months, even years, to get the complete picture.
If we have DNA sequencing data available, once the sequencing run is done, we can process it through our bioinformatics workflows and be able to provide you all that information in a matter of hours.
In a recent study we achieved very high accuracy and also precision in predicting whether a particular TB sample was going to be resistant to a particular drug or not.
And now we've got past that comparative study and we're demonstrating the performance that we needed to have. And therefore the decision was made to commit Public Health England to implementing it.
Science is day after day of fairly menial grinding work, collecting data, cleaning it, making sense of it, and then every now and again you get an extraordinary high when you look at the results and you see something new and you realise you're the first person in the world who's ever seen this.